about this product from
Yep.
Arbor Vitae, which is really Juan Arellano's one stem cell shot, is that they have a barcode and a QR code on the bottom. That's pretty cool. So I'm jealous of that, and I think that we need to be able to individually label every tube. It's been a challenge because if you try to do something, you know, you can't really write on every single tube. It's not scalable. So we put things in a pouch and we label the heck out of the pouch. But this is really cool. It does slip on and slip off. I think ideally we would be printing straight on the tube. The other thing they got going on here is their lid is set up for robotic capping and decapping and all that kind of stuff. Now, whether or not they actually use that aspect of the lid, this is actually a really nice feature. We don't have that on our lid. That said, this tube doesn't hold 2 mils. So it's shipped and sold as 2.2 cc's. That's the claim, the verbal claim. I measured what was in the tube. It was 1.7 cc's.
Okay, and he told me he always puts extra in all of his,
yes,
his vials.
1.7 is what I measured in this tube. Now, I have several more to test, and I'll continue to test them. But if you look at it, there's no color, there's no particulate. It really turned out to be mostly saline. And we could go back and we can actually take it. I'll send you pictures of the slides. That's the easiest thing.
Perfect. Yeah, I'll link those to this.
The slide's really pretty much empty. There's nothing in this tube. Now, whether or not, like I said, I have several more to test, and I want to make sure that it's documented so that people can't accuse me of tampering with the product. So when we test the rest, we'll see if it's a consistency issue, because that's actually really, I think, a challenge in our space is being consistent. I mean, every donor is different, every isolation, is different. Maybe you have different technicians and so on. Now, we address that problem by normalizing based on live cell count. So every single lot that we produce, we count the live cells to determine how to normalize that. And so in our objective there, of course, is to make sure that the physicians can establish protocols and expect similar outcomes based on a particular protocol. That's the number one reason for doing that.
Great.
So I think that's really critically important. If there's a consistency issue, then that's something they can easily fix in-house. However, if every single one fails to produce any live cells, well, then there's some dishonesty on what's being, marketed.
And one thing, Eliott just showed me. Hi, everybody. Eliott just showed me the studies he did. Do you mind if we show him your machine?
Yeah, absolutely.
And just looking at viability and just trying to say, okay, well, what's really in this product? How does it look? Can you explain what this little gadget is?
Yeah, so this is actually a microscope connected to a camera that you use in conjunction. It's a kit that's pretty easy to use. Anybody can use it, actually. You use these slides that look like this, and you load them up with a sample of the tissue or product that you're trying to count the cells in, and the product's been stained with acridine orange or propydium iodide. Those are 2 dyes. One dye will go inside of an intact cell nucleus and only there, and stain that nucleus, and they show up as green on the imagery. The other one will only stain unraveled and dead DNA from a broken cell, a dead cell. Okay. And so it's a really great way to then contrast that with the bright field microscopy, where you get actual cell bodies on there. You can count them in whole, and then you count the dead, and then you count the alive. And with all that, it does some math, and it spits out how many are alive and how many are dead, what the viability is.
Yep.
And so an example of the Arbor Vitae product, or one stem cell shot, Juan Arellano product out of Texas, at least it appears to be from there, is, and like I said, we have some more we want to test so that we can actually freely distribute this information, because we really need to clean this space up. I'm excited when somebody makes a really good product, but not when someone claims to have made a good product and then actually be completely dishonest about it. It's bad for the industry, it's bad for the patients, it's bad for regenerative medicine in general. Okay, so this is an initial preview of a slide of Arbor Vitae product. Notice that there's really nothing on that slide. Normally there would be green spots wherever there's a live cell. We're just going to count this puppy the primary cell lines, AOPI, acridine orange propydium iodide, and then the going to choose the slide that we created. This is from a few days ago, but it took pictures, and now it's doing all the calculations off those pictures. The viability, the viable cells is 0. There are no living cells that this machine could detect. The total number of cells, because it's counting live and dead cells, is 3.8, 38,000 cells per ml. So in every cc of this product, you have 38,000 total cells, but they're all dead. So let me.
1.7 mLs is what you were using. Is that the same quantity?
So I just used actually just a small sample of this, 20 microliters, mixed with the dye.
Got it. Okay.
So it's a sampling. But here's what is interesting. It's also useful to go in and look at the slide itself in the bright field. So you're not even looking at the dye. This is just what the naked eye could see if somehow you were able to zoom in and turn your, look through a microscope directly.
Yeah.
That is not a cell.
Okay, it's squiggly line.
I don't know what it is. Okay. I was concerned enough about that. It looked like a contaminant. That I actually took this product and I plated it out to determine if there was some sort of a, bacterial fungal contamination that would then grow on the plate, and there was not. Thank goodness. So to be fair, this did not trigger any of those kinds of concerns. But that is not a cell. Who knows what it is. But if you look at this slide, it's pretty empty, but you see little things that are going to be counted as cells here and there, but they all happen to be dead because the dyes basically are only adhering to the dead and broken nuclei. So that's what that slide looks like of that product. But we have more to test, and I think that to be fair, you can't just test one unit. You should test multiple units of anybody's product if you want to really determine whether it's a consistency issue or, something like that. Consistency issues, I think, are easily remedied, but if there's never a living cell, then it's a hard uphill battle to solve those problems. And it's challenging to have living cells post-thaw with solid tissue-derived products.
And so can you show a contrast of
your
product
like?
or another product?
Yeah, absolutely.
I mean, because I agree. I think, not to talk bad about other labs or anything like that. It's just we need to know the truth, and
we need to be transparent.
That's where I came to you originally, as I said, look, I just need someone to shoot straight with me about what's in because what we tell our patients is so important, and if there's no viability in these cells, then we need to start saying, well, it's only proteins, which is what Juan was telling me. There's 62,000 proteins or something that he's isolated in there, and I can even count that. I mean.
No, that's not.
I was like, how many are in?
You can put a price tag on that, and it's something that I don't know anybody could afford, because the whole scientific community, in aggregate, has not done that yet.
Yeah, okay.
They haven't identified all the proteins and measured them and so on. And there's variability. We know from donor to donor, we get all sorts of variability, unfortunately. So we have to find an axis to normalize on. And right now, the way we're choosing to normalize is based on live cell count. But in reality, there are so many different things in this product. It's not a single, molecule like a lot of drugs are.
Yeah. And so what have you done to stabilize yours? What would you say like the main components are?
Well, so we have done things to stabilize them to the point where we've been able to, and this is obviously something that we're not selling. This is research and development type of thing. But we've been able to actually grow cells here after processing them into a flowable state. So they're flowable, they're ready to go into a microvial and ready to ship out. But instead of that, we've decided to see how these cells are looking, because one of the big challenges, this is a picture of one of our lots, one of our products.
Okay, so lots of green dots.
Lots of green dots, those are live cells. If we want to count these, we just count them real quick and it'll tell us what we got. And if you told me, 3 years ago, that 50% of the cells are going to be dead, I would have been pretty depressed. But the reality is that no one's even come close. I mean, we're talking about our next competitor is 3% potentially, and that's in the margin of error. I mean, these are really vibrant, living cells. And what that says, we're not talking about efficacy. We're not saying that it's the living cells that are efficacious. What we're talking about is the craftsmanship of being able to take the solid tissue and turn it into a flowable, and to preserve as many of the things and retain the integrity of the initial source material, as well as this. And
Got it.
so far, apparently, surprisingly enough, this is state of the art. So we have not found much competition here. So,
and this is Arnold Kaplan went at the perinatal stem cell conference. He threw out, well, dead cell or live cell, they still have healing. Yeah, there's
growth factors, etc.
Yeah.
Absolutely.
So what do you think?
So if I were a doctor in today's marketplace and I were looking for the best product, I would want to look at density of the product, and you could do that with a simple microscope image. Just look at density. Make sure you understand and trust whoever's talking to you about it, the source, that they have actually sourced it from X, Y, and Z, so you're not wondering where they got the cells. And then look at the density of the product, because almost everything in the marketplace today is dead. It's all dead. Centeno was right about that one single thing. and so
the professional blogger, is that who you're referring to?
The guy that's protecting his industry through fear, uncertainty, and doubt. So the rather dishonest journalism, in my opinion. So I wouldn't even kind of call it journalism, but he's right about that. So they're all dead, and the challenge here is that they're dead going in. And if they're not dead going in, they're dead as soon as you thaw them out.
Yep.
And that's been the understanding and what we've observed here in our lab with testing other companies' products. The cells are dead or dying. And so I've actually loaded up a slide before, case in point, where, and I have, control slides where this is not observed, but when I load up a slide post-op of a competitor product, there may be 3% living cells. But if you count again in 30 seconds, they're all dead.
They're gone. Okay.
They're gone. They're dying that fast, is kind of what I'm saying. So, and that's a challenge. But we've been able to, in-house, in our R&D lab, I have to say, because we're not doing any sort of cell expansion here, we've been able to actually expand the cells in the refrigerator with the different techniques that we've developed over the last 2-3 years to actually take the cells from an apoptotic state, a very disrupted state, an unhappy state, post morselization, flowability, and turn them into something that's stable, that's now either growing or remaining static.
Great.
And so that's unique, and so far we haven't found anybody that's done that.
And so as far as, when a cell's undergoing apoptosis, they influence all the cells
That's
around
correct.
them. And so
apoptosis is like, consider it, it's like a stack of firecrackers. First, you already started the first wave of death by taking these cells and mechanically taking them apart from each other so that they flow through a needle, which is the end goal. The next thing that happens is those cells that you've killed signal the neighboring cells to undergo apoptosis. And that apoptosis is then, of course, has another cascading effect. So it's like lighting, a whole stack of firecrackers.
they all just start going off.
They set the other ones off.
And so how have you stopped the apoptosis so that when we thaw out the tissue once it arrives at our clinics,
right,
so that we're not just, it's not like a bunch of fireworks going off all at once?
Yes, exactly. So it's a great question, and I would love to be able to answer it in detail. But if I were to have answered that in detail 2 years ago, these various other folks who've come in to my lab,
got it, yeah,
walked in and out the door, would be doing the same thing, but it would be in a way that wouldn't be good for the marketplace.
Okay.
I say we keep pushing the science forward. I think our lab's going to grow. We're going to have more scientists here, and it'll be a better sort of collaboration and growth. And I hope eventually to be able to talk to some of the leaders in this space and try to share ideas and and see, where we can go next.